What's up, everybody? So in this video we're going to be talking about chemical signaling, everything that you need to know about chemical signaling. When you hear the word chemical signaling, you probably shut down and be like, oh, that just sounds complicated. But to be honest, it's not so bad, right? So there's a few things we got to cover, and this is actually a pure HL topic. SL doesn't do C2.1, so there's not that much, only HL stuff for you guys to cover. So we'll get into it just now. So remember, guys, teachme.org is waiting for you. Really really sexy notes and lots of IB style questions that you need to prepare for those final exams. So anyways, guys, let's get started. So in a nutshell, cell signaling is actually really straightforward if you think of it like cell communication. Me and you, in our everyday life, we have to communicate with our friends and our family members and teachers, right, to make sure that they understand what we want and that we understand what they want. In that way, everything goes smoothly in society. No one gets upset, no one gets angry, and everything goes well, right? If we don't communicate, everything goes wrong because you thought they meant this and then you ended up doing that which upset them or whatnot, right? Communication is key. So we're going to use this little analogy here. Maybe you guys recognize it. This is the house from the movie Up. If you're an Up fan, you'd know that, right? So here's our little house, and probably the balloon gave it away. Now, each house, right, has a mailbox, right? A mailbox. A mailbox is the little thing to which the mail or the letters or whatever mail is going to be put into, right? It's a receiver of our mail. Now, the thing is, the mail is put into this mailbox. Now, the people inside this house are gonna go and get this mail, whether it's the old man, the old grumpy man, the overhyped kid, or the sad little dog, right? Whoever it is, they'll go get the mail, and whoever ends up reading the mail will end up doing what it says. So maybe this mail is destined for this old grumpy guy, telling him to pay his taxes, pay his water fees, or his electricity. So anyways, whoever ends up whoever the mail was meant for will get the mail, this message, and do what it says, right? So that's how it works in society, and that's communication. Someone sent him this message, he read this message, and he's doing what it says. Our cells work so similar. Let me show you. So here we got our cell, our cell that is going to receive the message. You can think of it like this little house, right? This house with people inside it or whatever. Now, this cell, just like our house, has a mailbox. Okay, what do we call this mailbox? We call it a receptor. A receptor, because it receives things, a receptor. Now, this receptor, how we can define it is a molecule that is going to bind a mail, okay? And in science, the mail we call a ligand, okay? So it's a molecule to which a ligand will bind to, okay? As you'll see later in this video, a receptor can be located on the membrane of the cell or inside of the cell, okay? So, but don't worry about that for now. Just pay attention here. Now, a ligand, okay, a ligand is going to be our mail, okay? So ligand is a little molecule in our body that's going to go and bind to this receptor, okay? Okay, normally this receptor is a protein, by the way. Remember, a protein is made up of a chain of amino acids, right? So normally receptors are proteins. Now this ligand, which is going to be our mail, or this message, is going to go and bind to this receptor. Once this mail is in the mailbox, now the house can go and read that message and do what it says. The same way with the cell. Once this ligand has bounded to this receptor, the cell will now be able to read that message and do what it says. And a whole bunch of reactions are going to happen inside of the cell. Okay, that's called our cellular response. Whatever this ligand told it to do, it's going to go and do now. Okay, so you can imagine that this cellular response is going to be a bunch of little steps to carry out the task. Okay, so we call those individual steps to carry out the task a signal transduction pathway. Okay, signal transduction pathway is going to happen after the ligand binds the receptor. Now you can imagine, without a mailbox, how are we going to receive the mail? How is the house going to carry out everything? So you can imagine the same thing with a cell. If you have a cell that has no receptor for this ligand, okay, so say it has other receptors, but maybe no receptor that's going to bind to this ligand, then that cell will not do what that ligand wants it to do. Because you can imagine, when you make a molecule, you don't want every single cell in your whole body to do that thing. You only want some cells to do it, the cells that are destined to do that function. So not all cells will have every single receptor, okay? So if there's no receptor, there will be no response. You need a receptor to be able to bind that ligand so that a response can happen. So that's really the whole cell signaling idea in a nutshell for you guys. So now we're going to go into talking about the specific kinds of ligands. We know ligands in general are these little chemicals, these male molecules that are going to bind to a receptor and cause a cell to do something. But we need to now learn about different kinds of ligands, categories of ligands, okay? So here we have our cell, okay? A very basic cell. I didn't put any organelles here. It's just to keep it simple for you guys. Now, in general, ligands can be classified into two categories. Okay, hydrophilic ligands or hydrophobic ligands. Now, this is really key. Hydrophilic ligands are the most common, MC. I just put it here for most common. Now, a very key thing you guys need to understand is, remember, our cell is surrounded by a plasma membrane, right? Which is the biphospholipid layer, right? Now remember, hydrophilic things, hydrophilic meant hydro means water, right? And philic means loving. So there are ligands that are hydrophilic, that like water, cannot cross this membrane, right? Because they can't get through that whole area that's hydrophobic in the membrane, the fatty acid tails of the phospholipid bilayer. They can't get through that because they're hydrophilic. So these ligands, they will bind to these receptors on the surface of the membrane. We call them surface receptors, okay? They cannot bind to the ones on the inside, called intracellular receptors, okay? Now, on the contrary, the hydrophobic ones, those that hate water, they can pass through that phospholipid bilayer because they can mingle with those fatty acid tails that are hydrophobic. So they can come in, and therefore they can bind to these intracellular receptors, okay? Those two are really important to understand because they love to ask you which can bind to intracellular receptors and which can bind to surface receptors. You should know hydrophilic ones can bind the surface ones and hydrophobic ones can bind the intracellular ones, okay? Now, there are four kind of categories of ligands. So ligands, right, are all these molecules that are going to bind to the receptor. We can classify them into these four vague categories, okay? Neurotransmitters, hormones, cytokines, and calcium. But I want to point out that if you had time to only learn these two. They're really important. I don't think they're going to care much about these two, honestly, based off my experience. Neurotransmitters and hormones, though, you guys got to nail these. So one category of ligands is neurotransmitters. Like the name says, these are the little molecules that transmit signals between our neurons. If you guys haven't learned about neurons yet, you guys will later, but those are the little nerve cells, right, that comprise our nervous system, these little freaky looking guys. And they communicate with each other by sending each other little messages, right? If you want to transmit our signal from this neuron to this one, you got to send a neurotransmitter that will then bind to this neuron and activate it. And then eventually this neuron will send another neurotransmitter to our muscle cells to cause them to contract. So these little ligands called neurotransmitters, they are one kind that you need to know about. Now, neurotransmitters can be any kind of structure. They can be proteins, like remember peptides are short little proteins. They can be amino acids. They can be nitrous oxide or amines. So neurotransmitters don't have one fixed structure. You have different kinds of neurotransmitters in your body, all with different structures, but they all have the same function of sending signals between neurons or between neurons and the target cell, like a muscle cell or a gland. Because, right, you need to activate your salivary glands to secrete enzymes to digest your food. Neurons can even send little signals to your salivary glands, okay? So what you need to understand is that the source is from a neuron, okay? They come from a neuron like these. And the target can vary. It can either be another neuron that's right adjacent to it, or it can be a muscle cell. It can be a target cell like a gland that's going to make enzymes to digest your food, okay? But the big thing they have in common is that the distance that they act is very, very short, extremely short, right adjacent. It's like you're able to touch your friend and tap them and give them a signal, right? It's the same thing here. Neurotransmitters basically tap on the shoulder of their adjacent cell, letting them know something. That ligand, that neurotransmitter, does not travel a long distance before it acts. It's very short distance. That's very important to understand. And the action of this neurotransmitter will vary. It can, for example, if the signal is sent, this neurotransmitter is sent from this neuron to that neuron. In that case, the action was to send a signal between this neuron and that neuron. Whereas another action could be to activate a muscle cell, causing it to contract. So the action will depend a little bit, depending from which cell the signal was sent and the target cell, okay? But the big idea, again, to get out is the short distance idea, okay? Very, very key. So here's one example of a little neuron that binds a receptor, and that's going to now cause this cellular response, okay? Cool, that's it for neurotransmitters. Really, really important. Now let's get on to hormones, okay? Hormones. So in the same way, hormones is a broad category. Hormones can be amines, they can be proteins, they can be lipids. So they can be various kinds of structures. It's not one set structure. But all hormones have the same kind of action, okay? Unlike neurotransmitters, they travel a long, long distance. Okay, let me show you here. They are released from whatever cell that makes them, and they travel a long distance in the bloodstream. Let me show you here. So if we have a cell that makes hormones, by the way, cells that make hormones, we call them endocrine cells. Endocrine is the science way of saying hormones. So these cells can be anywhere. Let me show you here a little diagram. Your whole body has so many little organs called glands that can make hormones, different hormones. For example, your pancreas, right? It can make insulin. Your adrenal gland can make adrenaline to make you feel that rush. Your testes can make testosterone. So you have many different areas in your body, little glands that can make hormones. So anyways, the cells that make hormones are called endocrine cells. And they, unlike the neurotransmitters that sends it directly to the adjacent cell, these guys, when they make their hormone, they put it into the bloodstream. So you can see here, this is our circulation here, okay, our bloodstream. And the bloodstream will carry it to a distant site, wherever that may be, depending on what the hormone is, okay? Now the hormone will be carried to the distant site. Now remember here, very key, so the source is the endocrine cell. Now a hormone can be either an amine, a protein, a lipid, or whatnot, okay? So depending if it is hydrophilic or hydrophobic, it can either bind to the surface receptor or an intracellular receptor. So let's say this hormone is a lipid. We know lipids are what? Hydrophobic. In that case, this hormone will leave the bloodstream and go through the cell membrane and bind to a receptor on the inside of the cell called an intracellular receptor. But if it was, let's say, a protein, by the way, I can't get over it. These guys look a lot like mushrooms. It was not the intention. But yeah, if this hormone was a protein, then it may be hydrophilic. In that case, it would rather go and bind to a surface receptor, okay? So it really depends if that hormone is hydrophilic or hydrophobic. But either way, it will bind to its receptor and it will cause a response, depending on what the purpose of that hormone was in the first place, right? That's key. So we need to quickly look at an example of a hormone, okay? So because hormones can have so many actions, right? It depends what hormone we're talking about. Is it testosterone? Is it estrogen? What is the exact hormone? So we're going to give you one little real life sort of example, but I don't want you guys to worry too much about this because we really cover this concept of like female reproductive system in a lot of detail in D3.1. So just listen to this part. Don't worry too much about taking notes or trying to really understand the hell out of this, okay? Just try and get the concept here. So we know this right here, right? This is the female reproductive system. This is the uterus. And this is the fallopian tubes connected to the ovaries, right? These are like the balls of females, right? Males have testes. These are like the testes of females. They make like the female hormones, okay? Now, there's a close relationship between the female reproductive system and the brain. So your brain, right, come back in this picture, has these two little areas that make hormones called the hypothalamus. And below the hypothalamus, connected to the hypothalamus, is the pituitary gland. You can see here. In reality, if you Google pictures of a the pituitary gland, it looks a lot like balls. You'll not believe it, like a lot. But whatever, not important right now. Anyways, doesn't this guy look nice and cute here, sitting with his feet and little glasses? Now, anyways, the hypothalamus, what it basically does is it has these endocrine cells that make hormones, and the hormones that it makes will be sent to the pituitary that's situated right below it. And those hormones will tell the pituitary gland to do certain things. So in a way, you can think of the hypothalamus as the master of the pituitary gland. It makes certain hormones that controls the pituitary gland. So for example, one kind of hormone that the hypothalamus makes is gonadotropin releasing hormone, or GnRH. I'll put the word for you here because it is a mouthful for sure. You know, scientists love to make these names. But if you break the word down, it's not so bad. Gonad means your private part. So for example, in male, it'd be the testes, and in female, it'd be the ovaries. That's a gonad. Now, trophin means to grow, or to make, to stimulate, okay? So these guys, these gonadotropin releasing hormones, they stimulate the pituitary to make gonadotropin hormone, okay? So once it stimulates the pituitary, sorry, I'm getting ahead of myself here. So the GnRH will stimulate the pituitary to release these gonadotropic hormones. Now these gonadotropic hormones, they will go to the ovaries and stimulate them to make gonadotropic hormones. I mean, to make certain hormones that the gonads make. So in this case, for the female, the ovary likes to make estrogen. So estradiol is a specific kind of estrogen, the most potent type, the strongest type of estrogen. And then it also makes progesterone, right? But again, the key thing I want to say here is, look, every time this hormone is made, it's not doesn't act with the cell adjacent to it. It is put into the bloodstream and it's transported to the target. So the same here, when the pituitary makes these gonadotropic hormones, it doesn't act on the adjacent cell. It's transported all the way down from the brain to the ovary by the bloodstream. And then when the ovary makes these hormones are put into the bloodstream and they will have their effects. So in a nutshell, what does estrogen actually do and what does progesterone actually do? And again, this is covered in much more detail in D3.1, so just get the overall idea here. But we know estrogen is responsible for developing the secondary female characteristics like growing breast, widening of the hips, body hair, things like that, right? But it also plays an important role in the menstrual cycle, regulating the menstrual cycle and pregnancy. The same with progesterone. But we'll cover this much more later, okay, guys? So, but the key thing I want you guys to see here, also the ovary makes eggs. The key thing I want you guys to understand from this is that how hormones work, how they are released, they control things, they regulate things, they change your body in some sort of way by making some sort of effect happen, and they act over long distance. Okay, there are many other kinds of hormones like testosterone and other ones, right? So this is just one example of the way a hormone works. I just want you to see that concept, that's all, okay? So that's it for hormones. The key thing is distance site. Neurotransmitters, the key thing is short distance. Let's quickly run through these last two. I really don't think they're too important, so let's go quickly over them. Let's go to cytokines. Cytokines, here's our cytokines. Woo! It's like a party guy. This guy, okay, this ligand, when it binds to a receptor, it causes basically your body to do inflammation, okay, and proliferate. So for example, if you get infected with a bacteria, okay, your body wants to kill that bacteria, get rid of it quickly. If you get stabbed, your body wants to stop the bleeding, make inflammation to make that area heal well. These cytokines pretty much do that. They will bind to these receptors on white blood cells, like your immune system cells, or any kinds of cells that will then help heal your body, okay? And they will cause these cells to proliferate, become more and more and more, and cause blood flow to go towards the area that is damaged, okay? So don't want to talk too much about these, but essentially they stimulate inflammation and cell proliferation to attack that bacteria and heal up your body, okay? That's really, really all you guys should worry about that, okay? It's because it initiates this whole cascade of reactions that allow that response to happen. Now, lastly, our calcium. So calcium's interesting, okay? Because it doesn't bind to the receptor, okay? It is actually released when another ligand binds to a receptor. So let's say we have this random ligand here, and it binds to this receptor, this surface receptor. What happens is calcium can be part of the cascade of reactions. So when this receptor is bound and certain things happen, like chemicals are released now, a cascade is happening, calcium could be one of those molecules. We called that a secondary messenger. So it can now serve as a ligand and bind to other things inside the cell and cause reactions to happen. So because it is not the first thing that binds to cause this whole thing to happen, but it's rather the second thing, we call it the secondary messenger. Okay? And what can its job be? It can cause muscles to contract. It can cause a bunch of things to happen, but don't worry too much about it. Just focus on knowing that it can be involved in muscle contraction. And it's more of a secondary messenger, not the main ligand. Okay, so again, guys, that's it for our ligand idea here. Please focus on neurotransmitters and hormones. They're the key ones, okay? Key ones. So now that we're done with ligands, we're going to talk now about the little partner that they bind to, the receptors, okay? There are 4 kinds of receptors that we're going to learn about, okay? So we're going to take them one by one, try and understand them, and really we're only touching the surface. In reality, it's way more complicated than I'm going to teach it, so sometimes it just won't make perfect sense, but you know, that's how it is. Sometimes you need to memorize some things just to get by, okay? So don't blame yourself if you don't 100% get it. At this level, at the IB level, you can't 100% get it, okay? We don't go into that much detail. So hopefully we try to make some ways of making it fun and easy to remember, at least, just because you guys truly can't really understand it at this level, okay? So as you can see here, we got a lot of funny looking guys that we made here to help you guys remember this, okay? So remember, our receptor, our mailbox, is the protein or molecule to which our ligand is going to bind to. Now, the key thing we need to understand here is that receptors have a very, very specific structure, super specific. And they bind to a specific ligand. So one kind of receptor can only bind one specific ligand. It can't bind different kinds of ligands. It's made for only one ligand. So if that ligand is present, then the receptor can bind to it and cause a function to happen. It can't bind random ligands. It can only bind one specific ligand to which its structure is complementary, meaning it fits one specific ligand only, okay? That's very important to understand. Now, upon binding, so if that ligand, let's say the banana, binds to this receptor, it will cause a receptor to change shape on the inside, and that will cause a whole cascade of reactions to happen. Now, we can split our receptors into 2 kinds. We already briefly mentioned it before. We can have these transmembrane receptors, the one that's on the membrane itself. See, we made the membrane really thick here of this cell, okay? So this is a cell, and there's no organelles, just for simplicity's sake. And then the other kind of receptor that we can have is intracellular, so ones that are inside the cytoplasm or inside the nucleus of the cell, as opposed to being in the membrane, okay? So these are the 2 big categories. Now remember when we talked about this previously, we said that ligands can be either hydrophilic or hydrophobic. Remember, hydrophilic ligands cannot cross this cell membrane. So therefore, hydrophilic ligands will always bind to our transmembrane receptors, all of these in the membrane. Whereas hydrophobic or very small ligands can pass through that phospholipid bilayer and then bind to intracellular receptors. Okay, so these are the 2 kinds and the kinds of ligands that they bind to. Now, there are 3 kinds of transmembrane receptors that you guys have to learn about. So we made 3 little diagrams here to help you guys remember it. So the first kind we're going to learn about is this monkey guy here, okay, which is called our enzymatic receptors. Enzymatic receptors. These guys are proteins, right? Because most receptors are proteins that kind of span the membrane once. They are this little protein that will go through and span the membrane once. That's what we call it, a single pass. It doesn't wiggle waggle like this one. Look at this G protein coupled receptor. This one is a G protein coupled receptor, and we'll also learn about this one. But you can see it spans the membrane. It waggles through it like this snake. So we call this a multi-pass protein because the protein passes through the membrane multiple times instead of a single time like this enzymatic receptor. And then the last one we're going to learn about, this one you guys probably already seen in some other videos, is our chemically gated ion channel. This is also a receptor, and it's also multi-pass, like this G protein coupled receptor. It can wiggle waggle through it and form this kind of ion channel. okay, which has a receptor on it, okay? So these are the three kinds of transmembrane receptors that we're gonna learn about, okay? Now, transmembrane receptors have some things in common that we need to mention. You can see that all of them have a portion that is outside of the cell, and a portion that is inside the membrane, and then a portion that is inside the cell. Okay, so we can call the part of the receptor that is facing the outside of the cell the extracellular domain. And then the part that is on the inside here, the intracellular domain. And then the part that's spanning the membrane, we call that the transmembrane or intramembrane domain. We'll see about that one later. Okay, I forgot to label it here for you guys. But you can see they all have that. They all have this part that's inside and then outside and then through the membrane. Inside, outside, and through the membrane, okay? Now remember, these guys are proteins. I need to show you something here. The receptors, because they're proteins, the parts facing out is in water. Because remember, everything surrounding a cell is water, and everything inside a cell is water, and everything in the membrane is fatty acids, so there's no water there, okay? So you can imagine that this part of the protein, the amino acids that make up this part of the protein, must be hydrophilic. They must like water because they're in water. And this part here that's inside the cell must also be hydrophilic amino acids. And then this part spanning the membrane that is inside the phospholipid bilayer must be hydrophobic. So you can see that these receptors have different domains. Okay, domains are basically just parts of a protein that can move by themselves. Okay, that are independent of the rest of the protein. Let me, I write it here a bit better. It's portions of a protein polypeptide that folds independently. So these legs here can move separate from the arms, and the arms can move separate from the legs and all that kind of stuff. I just think of it like a little part of a protein that moves by itself, okay? That is independent of the rest of the protein. Now, so you can imagine that there must be domains that are hydrophilic, domains that are hydrophobic, okay? And that's common for all of these kinds of transmembrane receptors, okay? Now, the fact that they have these separate parts allows the outside part of this receptor to interact with little ligands from the outside and the inside to make reactions happen inside of the cell, okay? So all of these domains allow the whole receptor to do its job as well as possible. Okay, so we need to now tackle them one by one. So the first one we're going to talk about is here, this chemically gated ion channel. Okay, and we're not going to have a separate slide for this one because I think it's straightforward enough. For these, I made separate slides that we're going to talk about shortly in this video, but they're a bit more complicated. But this one, I think, straightforward. So we're going to talk about this one right here. So these guys, as the name implies, these are receptors that bind to a chemical or a ligand. And once they bind to this ligand, it will trigger this gate to open because these are chemically gated ion channels. So ions are supposed to flow through them, but normally this thing is gated, it's closed. But when a ligand binds to it, or a chemical binds to it, these gates will open and the ions can then flow through. Okay, so one example you guys should know about of a ligand is acetylcholine. Okay, acetylcholine. I actually want to come back to this. Look here. So remember, acetylcholine is an amino acid. So, for example, this ligand maybe is a neurotransmitter, okay? Because you know neurons, let me come back to it here. Neurons send signals to each other, right? So, for example, let's say this neuron is sending acetylcholine between this neuron and that one. And when this acetylcholine binds to the receptor of this adjacent neuron, it will cause it to open and allow sodiums to come in. So the signal is then thereby sent from this neuron to this one, okay? So that's one example of a kind of receptor you guys should know about. Chemically gated ion channels, okay? So some ion channels can let sodium pass, other ones can let potassium pass or calcium pass, okay? There are many different kinds, so it's a very broad category, okay? Make sure you understand that. Okay, cool. So now, guys, let's get into the next one. So that's a simpler one. Let's now go on to G protein coupled receptor, this nasty snake here. So G protein coupled receptor, what a freaking name. Let's break it down first. So G protein coupled receptor is a receptor, okay, that is coupled to a G protein, okay? So this is the receptor. You can see here's the mouth that's going to bind something. So this is the receptor, and it's coupled with a G protein. So this here, these A, B, and gamma, alpha, beta, and gamma, this is the G protein. So it's a G protein coupled receptor because it's a receptor that's coupled with a G protein. Now, why on earth is this called a G protein? Because these G proteins, they bind to GDP. So here's GDP, okay? They always bind to GDP. So that's why it's called a G protein coupled receptor. It's a receptor that's coupled with a G protein, and it's called a G protein because G proteins bind to GDP. Okay, now how do these guys work? These freaks of nature. So let me show you. So there will be some sort of ligand, right? So let's just make it a random ligand. Now this ligand will bind to a cell that has this sort of receptor, okay? And again, this is a broad category. So remember here how I said that chemically gated ion channels, this is a broad category. There are different kinds of chemically gated ion channels, some that allow sodium, some that allow potassium or calcium, whatever. So the same here with G proteins, coupled receptors. There are different kinds, okay? But I'm gonna now explain how they all work in general, okay? But there are different kinds. Now in general, how G protein coupled receptors work is there will be a ligand, and then this ligand will go and bind to this receptor, okay? This receptor, this G protein coupled receptor. Now, once it binds to this receptor, okay, the tail on the inside, so the intracellular domain, because remember, this is the extracellular domain, the intracellular domain will move. And when it moves, okay, this G protein will move. And when this G protein moves, this GDP is converted into GTP. Let me show you. GTP. Sorry, let me fix something. I'm going to bring this to the front because right now it's hidden behind everything. So here, the GTP is now activated. So initially it was GDP. Now when this ligand binds, this tail will move and the GDP on the G protein will be converted into a GTP. So I have the words here for you guys. Now once the GTP is there, and bear in mind, this GTP is always on the alpha aspect of this G protein because there's 3 parts to this G protein, right? The GTP is always on the alpha part. So now when the GTP is there on the alpha part, now this alpha part can dissociate, okay, with the GTP. Okay, so now it separates itself from the rest of the G protein. Now when it does this, this alpha part can migrate and it can go and do various functions, okay, depending on what kind of G protein coupled receptor we had. So it can either go and bind to these ion channels and activate them. It can go and bind to some other enzymes and activate them, okay, whatever. But all G proteins work the same till this part, okay? They all work the same until this part is dissociated. Now, a different G protein will have a different effect. So maybe some G protein coupled receptors, this part will bind to an ion channel. In other scenarios, it may bind to an enzyme or whatever, or even any other effect, okay? But they all work the same up until this point, okay? So you can see a G protein coupled receptor acts indirectly on enzymes, this is this donut, or ion channels with the aid of this G protein, this protein called the G protein, okay, which was here. So let's summarize real quick this mechanism, and then we'll look at a real example, because this is just an overview of how G protein coupled receptors work, but we didn't give you a real ligand or a real example yet. So we'll look at that one, a really cool example that's really interesting to learn about. So here's the summary, right? Ligand binds to the G protein coupled receptor. The receptor is activated and there's a conformational change. The tail will move, and this movement will cause the G protein to become a GTP on the alpha subunit. Once there's a GTP on the alpha subunit, this alpha subunit will dissociate. And then once it's dissociated, now it will do whatever function this one wants it to do. Okay, this ligand wants it to do. Okay, so that's general. It's really quite straightforward. Okay, now let's look at a legit example, guys. A really cool example. So here again, we have our membrane, the inside of our cell. This is the outside of our cell, and this is the outside of the cell on the other side. Okay, so here's our cell. Okay, so I know it's a really odd looking cell. It doesn't look like it's going to be round based off this trajectory of these membranes, but whatever, deal with it, guys. So we're going to look at a real example of epinephrine. Okay, epinephrine. You guys know epinephrine? It's adrenaline, the little molecule called adrenaline. They're both the same thing. One is Greek, one is Latin. The epinephrine is the Greek one, and then the adrenaline is the Latin word. But anyways, you guys know what adrenaline is. It's the thing that makes you, well, it's released when you're under stressful scenario, okay? It's like the fight or flight hormone. So let's say here, our epinephrine, right, is going to come and bind to any cell that has this specific receptor. Because remember, this epinephrine is only bounded to a G protein that is the specific shape for this specific epinephrine. Maybe another G protein that won't fit this epinephrine, and it will only fit another kind of ligand. Okay, so this ligand, this G protein, must be the exact complementary shape to this ligand, in this case epinephrine. Otherwise, no reaction will happen. Okay, so anyways, let's say they're matching and this epinephrine comes and binds. What happens? The same idea. This alpha subunit, this inside will move, and this GDP will become GTP, and it will dissociate, right? And in this case, for epinephrine, this little subunit will not go and bind to a ion channel. In this case, it will bind to an enzyme, okay? And this enzyme has a specific name you guys should know about, adenyl cyclase, AC. Sometimes they call it AC, okay? Now, and I made it a donut because cyclase means circle, okay? So adenyl cyclase, it will bind to adenyl cyclase. Now this enzyme, because it ends with an ACE, is going to convert ATP, okay, so there's some ATP inside the cell, into a cool molecule called CAMP. So I put a little tent here to remind you guys of that. CAMP. It's CAMP, cyclic AMP. Now this is a secondary messenger. Remember, guys, how I said earlier that calcium can be a secondary messenger because it's the molecule that's like released after the main ligand binds. It's the same way that calcium can be a secondary messenger here. You can also see that CAMP is not the main thing that bound, but it's released on the inside. So we can call this a secondary messenger as well. Okay, it's released after the main thing bound. Now this CAMP is going to do its own thing as well. This CAMP is going to now activate this kind of thing. By the way, from here, I don't want you guys to worry about the individual steps. It's in your book, but for a fact, you guys only have to understand the summary mechanism that I'm going to write here, okay? But I want to show you the real thing just because you understand the concept. But don't worry about the individual names, although it is in your book. So CAMP will now activate pKa, which is just a kind of enzyme, into pKa, activated pKa. So non-activated pKa into activated pKa. Now, so you can see, I want to emphasize to you guys that it's just after this whole thing binds, there's a whole cascade of reactions. That's what I want you to understand. Not the individual things that happen, that there's a cascade of one thing activating another thing and that thing activating another thing until the final thing happens. So now this pKa, guess what? It's going to activate another molecule into another activated thing, okay? Just so you guys know, this molecule is called phosphorylase kinase, but again, don't worry about it. Now it's activated phosphorylase kinase. Now this guy has a specific job to break down glycogen. You know, glycogen is a accumulation of a bunch of glucoses aggregated together, a storage form of glucose, okay? Because each of these individual ones here are glucose, but when they are aggregated and stored together, we call them glycogen. So this guy here is going to kind of go, it's like a scissors, it's going to chop this guy up so that all the little glucoses can be released. Okay, so now the glucoses, once they're released, they can come out of the cell and be released outside of the cell into our bloodstream so that it can be transported to muscle cells and things that need them. Okay, because glucose is a form of energy. So under what scenario would we even need epinephrine? So say you see a freaking bear, it's about to attack you, or you're about to take an exam, or you're about to do your heaviest deadlift, whatever. This stressful event, whether it's emotional or physical, will cause your glands. So do you guys know what gland releases this epinephrine? This guy here, our adrenal gland, right? It releases adrenaline. So once this adrenaline is released, it's going to be transported. It's going to bind to every single cell that has this G protein coupled receptor that is specific to our epinephrine. So it's going to bind, whole freaking cascade is going to happen. You guys should remember everything that I'm going to write here, okay? Don't worry about every single little detail here. I just wanted to show you that it's a cascade of things. Now, the stressful event, guess what? It leads to your adrenal glands making this epinephrine, and then this epinephrine will bind to this G protein coupled receptor, and then we'll have this alpha subunit dissociated, which will then bind to adenyl cyclase to be activated and convert ATP into CAMP, and then there will be a few more phosphorylation cascades, okay? That just means that this one is going to phosphorylate this pKa to become activated, and this one is going to phosphorylate this one to become activated. A cascade is going to happen until finally our glucose is, the glycogen has been chopped up and glucose is released into the bloodstream to be taken to our muscle cells, which needs them for energy so we can fight off this bear or lift up this dumbbell or this deadlift, whatever. So you can see that's really cool. This is how our body communicates with each other to be able to get ready for this stressful event by sending a little ligand, a little mail to a certain cell to tell it, my guy, we need glucose, release some glucose. And then this whole cascade happens, and then the cell will release glucose. One very key cell that does that is our liver cells. They store a lot of glucose. This epinephrine will go bind especially to our liver cells and tell them to have this cascade and release all the glucose so that all the cells that really need them can have that glucose, okay? So that's really, really cool how our body interacts and communicates with each other, with how the cells communicate with each other to make this effect happen, okay? So that's what you guys should know about our G protein coupled receptor. So now guys, we come to our second last kind of receptor, the category called tyrosine kinase receptors. So I always like to think of it like this monkey. His name is Tyrone, okay? Tyrone, he's our tyrosine kinase guy here. And this guy is also a transmembrane receptor, a single pass. So the protein passes once through the membrane, not wiggle waggle like the G protein coupled receptor. Now, as I wanted to do previously, guys, see here, we got the extracellular domain, transmembrane domain, and intracellular domain. Now, the thing about this one, this enzyme, this is an enzymatic receptor. Remember, enzymes, they catalyze reactions and makes things happen, right? So this is a kind of receptor that's also an enzyme. And let me show you what I mean by that. So this enzyme is activated, and again, it's specific to a kind of ligand. Let's say this specific kind of tyrosine kinase, it binds to insulins. Maybe another tyrosine kinase will bind to another kind of ligand. Remember, the ligand has to be complementary to the kind of receptor. So in this case, a tyrosine kinase, it needs to bind twice to the ligand. So you can see it's got 2 feet. So insulin is going to be this little banana, okay? This little banana. Now the banana is the ligand, and so it needs to bind twice, okay, to 2 parts of this tyrosine kinase. Once 2 insulins are bounded, now this little monkey's hands, this enzyme's hands here, the intracellular domain will become phosphorylated. There's like a lot of phosphates inside the cell. And once this binding happens, these little hands can move and they will grab onto these phosphates, okay, to become phosphorylated. Now, remember, it's an enzyme, so it's going to try and activate other things. So now that it's got these phosphates, it's going to want to give them to other little molecules. And by giving these phosphates to other molecules, they become activated and they are able to go and do some kind of reaction. Let me show you. So let's say there's this molecule inside our cell, and this tyrosine kinase now bound to this insulin, and it grabbed these phosphates. Now it's going to want to go and put them on this guy. So it puts them on this guy and activates it. Now that this guy is activated with this phosphate, it's going to go and do a specific action, okay? Now, because we're talking about the specific example of insulin, what is going to happen in the specific example? What is insulin going to trigger? So remember, where on earth does insulin even come from? Remember this gland here? Let me show you real quick. The pancreas, the islets of Langerhans, they make insulin. And insulin is a little hormone that's supposed to, it's a little protein that's supposed to bring down our blood glucose. Okay, so once it binds, now this guy, he's gonna have to try and do something to help bring down our blood glucose. So what this guy does is he kind of stimulates these little vesicles that have these little channels on them, these glucose channels, and he stimulates them to go to the membrane. So when this vesicle goes to the membrane and fuses with it, now inside the membrane, we're going to have a bunch of these little glucose transporters. And once they're there, their job is to take glucose and bring them into the cell, okay? By doing that, we reduce the glucose in the bloodstream. We reduce the amount of glucose outside of our cell, and by dragging them all into the cell. Because by dragging them into the cell, we remove them from the bloodstream. So now our blood glucose is a bit lower. Okay, and that was the whole point of this. So let me show you here a mechanism. So you can see it's very much the opposite effect of our epinephrine. For epinephrine, by this G protein coupled receptor, we try to stimulate glucose to go outside the cell and increase our blood glucose. Whereas here, we're doing the opposite thing. We're trying to reduce our blood glucose by bringing glucose back into our cells, okay? So let's show you here the mechanism. So if your body has excess glucose, maybe you just ate a massive cake and you want to reduce your blood glucose by bringing some blood glucose into your cells, your body's going to release insulin from the pancreas. Then this whole thing will happen that I just said, okay, and ultimately release, causing glucose to be intaked into the cell. That's very important because if your blood glucose is too high, it can cause damage to your little arteries and your little veins and cause them to become very hard, and you can easily get strokes and things like that. So it's very important to maintain a normal blood glucose level. So that's why this is very, very important. So that's one example of our tyrosine kinase, okay? Let's go on to our last one, guys, our very last one. So finally, we get to a different kind of one, an intracellular receptor. So this receptor is not actually in the membrane, it's inside our cells. So let me show you how this works. This means you're going to have a ligand. This ligand that's going to bind to an intracellular receptor will have to be hydrophobic, like a lipid hormone or a steroid hormone, anyone that wants to pass, that's able to pass this phospholipid bilayer. Okay, so it passes this phospholipid bilayer, and then it's basically trying to find its receptor. So in this case, we got our coffee without cream. Okay, this cream is going to be our little ligand, and our receptor is going to be this coffee. And it goes and it finds that receptor and it binds to it. It binds to it, so now we have our receptor signal complex. Okay, and now once this is formed, this guy will move into our nucleus. Okay, so this receptor can either be inside the cytoplasm or it can already be in the nucleus. Either way, it's waiting for its ligand. Okay, but anyways, once this receptor signals complex forms, it's going to go to a specific gene that it wants to influence, right? Because if through protein synthesis, you go and through transcription, translation, you take a specific gene and activate it to make a protein, and that protein will have a specific function. In this case, this molecule is going to go and find a specific gene and activate it and say, my guy, we need you right now, activate you. So let's say in this case, this ligand wants to activate a certain gene. So now we form this receptor signal complex, it goes to this gene and it says, my guy, activate. And it makes this protein, and now once this protein is made, it can go and have its effect, okay? So you can see that this will affect transcription, affect our gene expression, okay? Maybe it's going to make more of some proteins, or maybe we're trying to make less of some proteins, okay? But either way, we're affecting gene expression to make a certain protein to be able to cause a certain response. So that's vaguely how this intracellular receptor concept is going to work. Finally, guys, we managed to get through that whole annoying thing, the whole ligands and receptors. We're now getting into another idea of something called positive and negative feedback. Okay, this one should be a little bit more logical and easier to understand. So you guys know, right? I just told you we have so many of these glands and things that make hormones and proteins. I mean, hormones especially, that are going to influence our blood sugar or our stress level or our hormone level or whatever. All these guys have little actions. And the thing is, these molecules are very not toxic, but very potent. They're very strong, and they can have a huge effect on our body. And it's really important that it's regulated. Remember the word homeostasis, the regulation of internal body conditions? We want to make sure that these hormone levels and blood glucose level, our body temperature, everything is at a normal level. We don't want it to go out of control. Otherwise, we will die very quickly, right? So our body has very interesting mechanisms to regulate it, including negative feedback and positive feedback. And we'll talk about exactly what this means now. Let's starting with our negative feedback. So since we talked about glucose so much, let's talk about it a bit more. Remember, I told you it's really bad if glucose is not controlled, because it can cause you to eventually harden all your vessels and cause heart attacks and strokes and many other kind of problems in our body. So we need to regulate our glucose. So how does our body do that? Through negative feedback. So look at this little diagram here. First, I want to say, what is negative feedback? So negative feedback is basically occurs when a condition is not in normal range anymore, and action is taken to return it to normal range. So I like to think of it like your body notices, oh no, something's wrong. We got to tell the body to fix it. So that's called negative feedback. something is negative is going on, we got to fix it. Something is not in the normal range anymore, we got to bring it back to normal range. It's negative feedback. Something went wrong, we got to fix it. So let me show you here. So let's say we have normal blood glucose and something goes wrong. Maybe this person has some kind of disorder that they now have low blood glucose. They went for a massive run. They have low blood glucose. They used it all up in their run. So what's our body going to do to return our glucose to a normal level? So what's it going to do? Remember our pancreas? I told you this little guy here. Let me go back to here. This guy. Okay, it's a gland and it's going to make these hormones. This little guy has little areas in it. Little circular areas, which we call isolates of Langerhans. Okay, little groups of cells. And these cells are responsible for regulating our glucose. Let me show you how. So in this scenario where we have low glucose, these isolates of Langerhans have two kinds of cells, okay, alpha cells and beta cells. Now in the context of low blood glucose, okay, we're going to have alpha cells. These alpha cells are going to release a little molecule called glucagon. Don't confuse this with glucose, okay? Glucagon is the hormone that's going to increase our glucose release, okay? So we have low blood glucose. This alpha cell is going to be like, okay, guys, I'm going to make glucagon. It makes glucagon. Glucagon is the hormone that's going to basically cause our cells, through cell signaling, to increase glucose release, okay? Very good. Kind of like epinephrine, you know, how we saw the example of epinephrine, how it caused glucose to be released and increased blood glucose. Glucagon works in a similar way, okay? So what about the opposite scenario? Let's say we have high blood glucose. Someone has diabetes and their glucose is high. What's our body gonna do? In normal scenario, if you don't have diabetes, your body will make, you have to remember the other cell here, the green one, this is our beta cell. The beta cell of the pancreas will release another hormone, which we just learned about, called insulin. Remember? Insulin, this one here. It's going to act through this mechanism to lower our glucose. So it's going to release insulin, and insulin is going to act through that cell signaling mechanism to bring our glucose lower back to normal. So that's very important. This diagram is super important. Now I want to help you guys remember this a bit, because right now I can assume you guys will be like, how do I remember this thing? I found an interesting little way. So you can think of it like this. The beta cells, what it's trying to do is bring our glucose back to normal. So it's trying to bring it below. Okay, so it's gonna, the beta cell releases insulin to bring our glucose below. Okay, and the alpha cell will release a hormone to bring our glucose level above. Okay, so when it's low, you have alpha cells to release glucagon to bring it above, back to normal. And then beta cell will release insulin to bring it below, okay, back to normal. So hopefully this can kind of help you remember it. I like to think of it like this because it helps me a lot, okay? So this is only one example. Your body's temperature is regulated through negative feedback and many other things. So this is just one idea. So understand the concept and this one example. Hopefully it helps you out. Now let's go to positive feedback. Positive feedback is the opposite thing. So when something is increasing, your body will have a response to increase it even more. Okay, so it's the exact opposite of negative feedback. We're bringing it further away from normal. So it's a feedback system that reinforces or amplifies a response. And a really cool one, an example of this is contractions during labor. So you know during labor, what happens is let me show you here. The baby is getting very big, right? It's increasing in size and it's pressing on the uterus, the wall, okay, stretching it. So you can imagine the pressure is increasing, okay, because this baby is squeezing on the wall of this uterus because it's trying to come out. Now, when this pressure increases, a signal is sent to your brain, okay? Specifically, guess where? Your hypothalamus. Remember the hypothalamus? This area controls a lot of things, right? So the signal is sent to the hypothalamus, and the hypothalamus, remember, it's like the boss. And it's going to send signals to it doesn't do anything by itself. It tells the pituitary to do it. So it sends a signal to the pituitary. Okay? Now pituitary will now make a little molecule called oxytocin. Let me show you. Oxytocin. Oxytocin, guess what this molecule does? Is it causes the pressure to increase even more by causing this wall to contract. So remember now, the baby, it was increasing, causing the pressure, it's pushing on this wall, causing the pressure to increase. Now instead of sending a signal and trying to decrease the pressure, in this system, we're trying to increase the pressure even more to get the baby out. So it sends a signal to the hypothalamus, then to pituitary. The pituitary makes oxytocin, and oxytocin causes this wall to contract and increase the pressure even more. Okay, so you can imagine now the pressure is increased even more. Okay, so this oxytocin causes it to contract, pressure increase even more. Because the pressure increases even more, a signal is sent to the hypothalamus, back to the pituitary, and even more oxytocin is released, so the pressure is increased even more. Okay, and eventually, guess what? The baby is gonna get popped out. So this is an example of a positive feedback. Something happens, some factor increases, and then we have a feedback mechanism to increase it even more. Okay, that's the opposite of negative feedback. And this is one example you guys should know: labor contractions. So finally, guys, we're at the last part here, quorum sensing, and then we'll do a couple of questions, IB style questions for you guys. So quorum sensing is actually something that happens, it's a kind of phenomenon that happens in bacteria. So it's a mechanism in which bacteria can alter group behavior depending on population density. So I'll show you what this means, okay? So say you have a couple of bacteria, and they can make a certain molecule. We call this an auto-inducer molecule. And this molecule, it has a function, but the thing is, at low concentrations, it cannot convince the cell to carry out a certain functions. It needs to be much higher. It's like when people go out to protest. If only a few people protest, nothing is going to change. But if a lot of people do, then maybe there will be a change. It's the same here. At low concentrations, this auto-inducer molecule will not cause the cell to do anything. But let's say these cells start duplicating, so their population density increases, okay? So say start increasing. Then this auto-inducer molecule concentration on the outside will be much higher than compared to before. Now it's like there's a lot of people protesting. So now there's a lot of this auto-inducer molecule. And when there's a lot of this auto-inducer molecule, now it can have an effect. Let me show you. So let's say this auto-inducer molecule at high concentrations, it can go into the nucleus of the cell, okay, and bind to an intracellular receptor. We learned about this. And when it binds to an intracellular receptor, specifically called LuxR, an example of an intracellular receptor, this complex will bind to a specific gene, which we call the Lux box. And it will cause and activate this gene expression to make that protein. Now that protein specifically is called luciferase. And this enzyme basically allows bioluminescence to occur, which is basically the process by which a living organism can make light. So why is this relevant? Because these bacteria have a mutualistic relationship, a symbiotic relationship with a kind of squid called a bobtail squid. Now this squid will ingest these bacteria, and because they ingest these bacteria, now these bacteria are in a safe environment where they can't get attacked by other bacteria or other chemicals. They're nice and safe inside the skin of this bobtail squid. So now they can proliferate and become a lot in a very easy amount of time, whereas before in the environment they would die very quickly and they couldn't increase their population very easily. But because of this relationship, they can easily increase their concentration, and by doing so, this effect can happen and they can start becoming light. They can start emitting light. And that's useful for the bobtail squid, because you can imagine if you are a fish under the squid and you look up, you will think that is the sun, not a squid. So in this way, the squid can protect itself against predators by behaving like sunlight. So that's really, really cool. And that's a cool example of signaling that happens inside bacteria. So a mechanism which by bacteria can alter group behavior depending on population density. The more there are of them, this behavior can start happening, whereas before it couldn't. So here are the step-by-step that I just talked about regarding that. So this is just one example of quorum sensing with bioluminescence, but it's not only with light that this works. There are other ideas as well. But for the IB, only worry about this light one is okay, this light example. Okay, let's do some questions, guys. Which of the following is a transmembrane receptor? Both G protein and enzymatic. The steroid hormone is an intracellular receptor. So the answer here is B. In response to high blood glucose levels, right? If it's high, we want to bring it below. So we're going to need beta cells, and beta cells make insulin to bring the glucose down. So it's going to be B. Last one here. Which of the following? Nope, jokes. I'm not going to do that one, guys. If you guys want many more questions, go to teachme.org to see lots and lots of IB style questions. And I hope this video was useful. I hope you learned something, and I will see you guys in the next one.